Hello everyone. In this presentation, we'll talk about the cell, my personal favorite topic. This will be more of a review, as I'm sure most of you have taken some sort of cell biology course already, whether it be an intro bio or in Molex Cell. So this will be kind of more of a review than anything. If you have any questions about this content, please feel free to reach out, or if you haven't taken any type of cell biology course, definitely reach out to me regarding this. So the cell. The cell is the smallest unit of life, and all living things are composed of cells. There are some major components that make up a cell, and this includes the plasma membrane, the cytoskeleton, the golgi apparatus, the mitochondria, the endoplasmic reticulum, the cytoplasm, the nucleus, and lysosomes. And we'll talk about each of these components in the next couple slides in more detail. So first we have the plasma membrane. The plasma membrane is a lipid bilayer that's made of two layers of phospholipids. So there's a top layer of phospholipids here and a bottom layer of phospholipids, and it acts to separate the external environment from the internal environment of the cell. And it's selectively permeable, means that it can allow things to come in, but things don't just readily rush in, and it's also not a hard barrier where nothing is allowed in or out. The plasma membrane is said to be a fluid mosaic model, meaning that all the phospholipids and all the proteins are not anchored in place. They're able to freely move around laterally throughout the plasma membrane. So it's kind of more of a dynamic, kind of almost like a vibrating type system rather than everything is kind of stationary. The plasma membrane has various proteins and macromolecules that bisected both on the external and internal side, and some that are integral that go all the way through. Some of these things are play a role in membrane rigidity. So in this image here, these yellow bits are supposed to be cholesterol, and that plays a role in how rigid or how malleable and flexible the membrane is. Also, there are many proteins and that play a role in cell-to-cell interactions. So proteins on one cell will interact with proteins on another cell, and that once they interact with each other, they bind to each other, it will cause a response in those cells, some sort of induced cellular response. They are also involved in passive and active transport. So some proteins, like the one that's visualized here, that's probably a poor color to use. Maybe I should use like a red. Yeah, there we go. The protein is supposed to indicate like a transport protein, where they allow certain compounds and molecules to come in, while others to leave. The membrane also plays a role in endo and exocytosis. So endocytosis is when you make a vesicle to bring something around something to bring it into the cell. Exocytosis is when you have a vesicle that fuses with the membrane to release something from the cell. Also, the membrane plays a role in ligand binding. In the environment, there are various macromolecules, various chemical signals that will bind to proteins and molecules on the outside of the cell and will allow for some sort of response from the target cell. And it plays a role in diffusion of certain compounds across the membrane. Next is the cytoplasm. The cytoplasm is a jelly-like material that fills the cell. So it's not like air or just water that's inside the cell. All of the internal components are kind of suspended within this jelly-like environment, including the organelles, vesicles, and various salts and macromolecules are also kind of suspended in this internal jelly-like substance. And then we have the cytoskeleton. So the cytoskeleton provides structure and support for the cell. It's basically kind of like the beams of the house. It also acts almost like the highway of the city, but we'll talk about that in a second. There are three types of filaments that compose the cytoskeleton. These are microfilaments, microtubules, and intermediate filaments, which are all kind of visualized here in this image. And they all play specific roles within the cell. The cytoplasm also plays a role in cellular locomotion. So if you've ever seen like an amoeba crawling or a cell even crawling sometimes from place to place, that is the leading end, the cytoskeleton is being constructed. On the lagging end, it's being degraded. So that movement, if it's not via flagella, if it's some sort of cell crawling, like in the case of an amoeba, or like macrophages and things like that, it's due to the cytoskeleton kind of pushing that cell forward. Or not pushing, yes, pulling that cell forward. And the cytoskeleton facilitates relocation of organelles and vesicles within the cell. So as I mentioned before, the cytoskeleton, depending on what filaments you have or what elements you're using, acts almost like a highway. So have you ever wondered, if is everything in the cell is still, how does anything move around? And it's via this system. So there are, if you look at the first image, that's actually a vesicle being transported through a cell, and that's via these filaments. The nucleus. The nucleus is the home of your genetic material, right? It's the brain of the operation. It is the most prominent organelle in the cell just because of its sheer size. It's pretty massive, and it's the site of transcription. So when you're trying to make proteins, of course, you go through transcription, then translation. Transcription occurs within the cytoplasm. Transcription occurs within the nucleus, and then translation generally occurs outside of the nucleus. The nucleus is encased in a lipid bilayer as well. There are two layers. The inner, which in this image is a bit hard to see, but the inner layer, which is here, and the outer layer, which is here. And the outer layer is actually continuous with the endoplasmic reticulum, which you can kind of see here. It keeps going and extends to the ER. And nuclear pores, which are these here, these little orange pepper looking things, they link the inner and outer membrane. So following that natural progression is the endoplasmic reticulum. Like I mentioned, it's continuous with the outer membrane of the nucleus. And there are two components to this. There's the rough ER, which is responsible for protein synthesis, and it's usually proteins that are destined to be embedded in the plasma membrane, the external membrane, that are going to be present in lysosomes, or proteins that are going to be exported from the cell. And then you have the smooth ER, which is responsible for lipid synthesis and cell detoxification. Then you have the Golgi apparatus. So the Golgi is actually more interesting than I think most people give it credit. It's responsible for post-translational modification of proteins and packing of proteins. So proteins that go from the ER are then packaged into vesicles, and they are shipped to the Golgi apparatus. And at the Golgi apparatus, on the cis face, you have proteins will enter the cis face of the Golgi via vesicles, and they'll get to this medial region. And in the medial region is where they'll undergo some sort of modification. This can be cleavage, glycosylation, phosphorylation, whatever kind of modifications are necessary. Proteins that need these modifications have no function, they won't function properly without those modifications. So this step is essential. They will then move, those proteins will then move to the trans phase, and once they're mature, so they're packaged into vesicles, move to the trans area, and after they move to the trans area of the Golgi, they will then go to their next destination, whether that be the lysosome, the plasma membrane, wherever have you, right? The next thing is lysosomes. So as mentioned, some of those vesicles containing proteins that leave the Golgi will then go to form lysosomes or fuse to existing lysosomes, make them a little bit bigger. So lysosomes are vesicles that contain a variety of enzymes, and they're responsible for breaking down unwanted material that gets into the cell. So this could be, as in the image here, it could be foreign materials from the external environment. They can be internal materials, such as things that have been brought in vesicles, things that are like damaged organelles, or anything else that's within the cell that needs to be destroyed. The lysosome fuses with a vesicle containing the target to be destroyed, and the vesicle will empty its enzymatic contents, and those enzymes will then chew up whatever is the target. Next, we have the mitochondria. The mitochondria is prokaryotic in origin, actually. If you go way, way back, at some point, a eukaryotic cell engulfed a prokaryotic cell, which was later to become the mitochondria. And there's proof of this because if you look at the mitochondria, it actually exhibits a lot of characteristics similar to bacteria, like having circular chromosome, circular DNA. So, but the mitochondria, as everyone always says, is the powerhouse of the cell. Yes, it is, but it does play a lot of other important roles within the cell itself. It plays a role in calcium storage, fatty acid synthesis, apoptosis, signaling, like producing cell signals, all that type of stuff also happens in the mitochondria. So it's more than just the powerhouse of the cell. It's actually pretty dynamic organelle. Alrighty, so some reading and resources. Read chapter 3, particularly chapter 3.1 and 3.2, and then the Smart Book assessment, as well as the video for on basics of cell biology. Have a good one.